Okay, here we are going to discuss the processes of RDT. So the processes of RDT or recombinant DNA technology. The first process is isolation of DNA. Okay. What is the isolation of DNA? So the isolation of DNA is the DNA that we are used as vector DNA or any type of DNA can be extracted out from their cell by using suitable enzymes. For example, suppose this is an animal cell and this is the DNA. So how can we extract this DNA from this animal cell? We are going to discuss here. So we are discussing here how to extract the DNA from the cell. So the DNA is extracted from the cell by opening the cell, by using suitable enzymes. Suppose there is a bacterial cell, then this cell can be opened up by using enzyme lysozyme, because lysozyme disrupts the cell wall of bacteria. So this enzyme disrupts the cell wall of this bacterial cell and we can easily extract the DNA from the bacterial cell. So lysozyme, this enzyme is used to extract the DNA from bacterial cell. Now if there is a plant cell, then we use enzyme like cellulase pectinase to extract the DNA from plant cell, because these enzymes, cellulase and pectinase enzyme, disrupt the cell wall of plant cell. So by disrupting the cell wall of plant cell, we can easily extract out the DNA of plant. So these enzymes like cellulase and pectinase are used to extract the DNA from plant cell. If suppose there is a fungi cell, then we use enzyme chitinase, because the cell wall of fungi is made up of chitin. Chitin is a homopolymer of N-acetyl glucosamine. So chitinase enzyme is used to disrupt the cell wall of fungi, and then we can extract the DNA from the fungi. So these are the enzymes which is used to disrupt the cell wall of bacterial, plant cell and fungi cell. So we can extract out the DNA from these cells. Animal cell does not have any cell wall, so there is no such enzyme for animal cell. Now suppose this is a bacterial cell and this is the DNA of bacteria. Then first we use enzyme lysozyme. This enzyme will disrupt the cell wall of the bacteria. Now we can extract out the DNA, but along with the DNA some macromolecules are also extracted, like RNA, protein. So these macromolecules are degraded by using suitable enzymes, like RNA can be degraded by using enzyme RNase, protein can be degraded by using enzyme proteinase, and other macromolecules are also degraded by using suitable enzymes. Finally the purified DNA is precipitated out by using chilled ethanol. This is important, chilled ethanol. Finally, after degrading the macromolecules like RNA, protein or other macromolecules with suitable enzymes, then at the end the purified DNA is precipitated out by using chilled ethanol. Then after using this chilled ethanol, DNA can be seen in a suspension as a fine threads. After using chilled ethanol, DNA can be seen in a suspension solution. This is suspension solution. This is suspension in the form of fine threads. This is DNA. So DNA is precipitated out by using chilled ethanol, and we can see the DNA in form of fine threads. At the end, this DNA can be removed out by using spooling method. Spooling method. Spooling is a method in which we use a glass rod to extract this DNA in form of spool. So what is spooling method? In spooling method, we use glass rod. To extract this DNA in form of spool. So this is the procedure of extracting out the DNA from the living cell.